Okay, so it's my pleasure to be introducing our first presenter for today. It's Dr. Ahmed Mostafa. Dr. Ahmed Mostafa earned his MD and completed his residency in diagnostic laboratory medicine before obtaining a PhD in immunology and infectious diseases from Memorial University of Newfoundland, Canada. He then pursued postdoctoral training at the University of Calgary and specialized as a clinical fellow in histocompatibility and immunogenetics at Alberta Precision Labs. Dr. Mostafa became a fellow of the American College for Histocompatibility and Immunogenetics. He is currently the clinical director of the HLA lab at St. Paul's Hospital in Saskatoon and associate professor in the Department of Pathology and Laboratory Medicine. Please join me in welcoming Dr. Mostafa.
Oh, thanks, Jenny, for this nice introduction. I didn't know that I'm all this positioned at the same time. So before I start, I would like to thank all the Vancouver team, the HLA lab, and the nephrologists and everyone for organizing this meeting and inviting me to present some of the work that we do in the other part of the country. And special thanks to Sari Noble for all this organization and very detailed emails that she sent to us was very helpful. So I guess we will start. Mouse. It's frozen. Okay. So before I start, I have a few disclosure here. So half of this work that I'm presenting today have been presented previously in different other meetings. So it was selected one of the top 5 abstracts for the 38th annual HLA transplant workshop. And Eric, my student, got this award and he presented this work orally. Eric also got a travel award to present the work in ASHI this year. In addition, the work was presented in the Canadian Society of Transplantation, and I was selected for the first place award. And in addition, it was also presented in the Transplantation Society in Istanbul. And actually, these are my Turkish friends in Istanbul, and after they listened to the talk, they were ready to implement epitope matching and using some of these techniques in their lab. So, as you know, our fields, actually like immunology in general is a very new field and HLA is more recent. So the story of HLA started in 1958 by the Nobel Prize winner, John Dessay, who was a French scientist. And at one time, in his lab, he noticed that people who get multiple transfused, they get some sort of reactions. So he asked the technician one time to bring him a blood sample, and he started to do some games, actually, in his lab. So what he did, he isolated the white blood cells. And at this time, they didn't know that white blood cells carry HLA on the surface. We now know, so that's why I put the HLA on the surface. And what he did, he started to get plasma from people who have been multiple transfused, from patients who have been multiple transfused. So now we know that these plasma contain antibodies. At this time, there was no information. And he started to put some drops from this patient to the white blood cells from the different donor. And he noticed that 2 observations. Sometimes it gets agglutinated and sometimes it doesn't. But what he actually interesting found that it never agglutinates its own cells. And the first 3 donors who donated the blood, their initials started with MAC, and that's why the old name of the HLA was MAC. But it got changed over the years. So the discovery of Dr. Jean Dessay has been used until maybe early 2000, which is we known as the CDC. So it was the gold method for typing, it was the gold method of cross-matching, and it was also the gold method of HLA antibody. So just one discovery. So the discovery also led us for decades to understand that or at least to know that there is some sort of HLA antibody that react with a specific antigen. So for example, here we can see that if this HLA bind to the A1, then this is called HLA A1 antibody. If this one bind to the DR7, then this is a DR7 antibody, and this one could be the CW9, and so on. But this we have known this for a very number of years until much more recently. We know now that antibodies does not bind to antigens. Antibody bind to specific parts of antigen we know as epitopes. So, for example, here it actually looks like the same idea of the key and the lock and the key. So, if the antibody properly fit to the epitopes, it will bind. And also, if these epitopes are present in different antigen, the same antibody will bind to the other antigen, which we know as cross-reactivity. So our hypothesis actually started by this simple diagram. I know that yesterday Karen showed us some crafts and how she teach the kids on how to understand epitopes, but I think this is much easier for us. We are not kids. So basically, if this is a donor who express B18, and this donor have 11 different epitopes, and I put these epitopes as different colors. If there is a patient who is B7, but this patient does not see this 11 epitopes. So if we go into transplant this patient, the risk that this patient will develop 11 different antibodies against these 11 epitopes. But if we see the whole picture, the rest of the typing of the patient may be B52 and A33, you can see that some of the epitopes that are in the donor are actually expressed in other antigens in this patient. So although the patient and the donor has one antigen B mismatch, but they have 0 epitope mismatch. So we think that this idea is more crispy in identifying immunological risk. And we started this project by recruiting 616 patients who has been transplanted in the province. And this was for the last 10 years. So we picked them from 2012 until we started this project in 2022. And we monitored these patients over the 10 years for developing of antibody-mediated rejection and donor-specific antibodies. And here, the first figure here represents the number of patients that have actually developed donor-specific antibodies. And you can see here, as we have seen from previous talks yesterday, that the majority of the donor-specific antibodies are directed against DQ. So in this example, there was like almost 60 patients developed donor-specific antibodies against DQ. The next one was the HLA-A, and then the DR. So the idea is very simple. We use the Matchmaker, which is actually built in the HLA Fusion software. And here is just like an example on how the software predict the epitope matching. So first you put the patient typing, and then you put the donor typing, and then the software will compare the patient and the donor typing. And in the next column here, you will see that the software will spell out all the epitopes that are different between the patient and the donors. And this is the difference. So if you've seen a number here, this means that the patient have the donor have 10 epitope mismatched than the donor for the DR locus, and 28 epitope mismatch for the DQ locus. And you can do it with every single locus in the HLA. So sometimes like a patient come to the clinic with 2 donors, and they ask the nephrologist, transplant nephrologist, which donor is the best donor for me. So the first thing, they look at the matching. So if the patient has one donor has 1 antigen mismatch and the other donor has 2 antigen mismatch, then the transplant nephrologist will tell him, oh, yeah, we will go with the one patient who have 1 antigen mismatch because they have less risk of developing antibodies. But let's see in this graph how we interpreted this result. So here you can see this is, for example, the HLA locus. And we divided our cohort into 2 groups. So The 1 and 2 that you see in the bottom here, these are the one group that have one antigen mismatch, and the number 2, these are the group that have 2 antigen mismatch. And we remove the group that has 0 antigen mismatch to avoid bias. And if you see here, you can see, like, for example, this small dot, this is one patient. And you can see this patient has 1 antigen mismatch, but have 20 epitope mismatch. And if you see this patient here, this patient actually have 2 antigen mismatch and have less than 5 epitope mismatch. So definitely epitope matching has a better, higher resolution risk assessment for antibody formation. And we actually did not only do it on the A, we actually did it in all the loci that we tested. And from this data and from the patient that developed donor-specific antibody, we were able to calculate the sensitivity over the specificity, and we were able to calculate a certain cutoff, because the idea here is we need to give the clinician a cutoff to tell them that above this cutoff, your patient is more risky than if it's below this cutoff. And based on this analysis, we were able to create different cutoffs for each different locus. So for example, here you can see like the HLA, if you have more than 15 epitope mismatch, you are at a higher risk than if you have less than 15 epitope mismatch, and so on. And you can see that this cutoff are different between loci, but mostly are between 10 to 15 epitope mismatch. And we never stopped at this point. We also started to monitor this patient over the time to see the survival of this graft. And you can see here, only for simplicity, I added the HLA-A and the HLA-DQ. And by doing this, we were able now to stratify this patient into 3 different groups. So for example, if we talk about the DQ, since it's more common donor-specific antibody, you can see that if this patient has 0 epitope mismatch, they are the best survival. If they have between 1 to 8 mismatch, they are medium risk. And if they have more than 9 epitope mismatch, they are definitely at a higher risk, even can proceed into graft failure. And after all that, when we talked to our transplant nephrologist, we were able to interpret all this data into clinical. So now, if you can see, this is our, for example, one of our group report. In the top, you can see that the patient typing, and in the bottom is the donor typing. And then you can see the number of mismatches at the last line here. So for example, if the donor is 24 and 68, then there are 2 antigen mismatch. But when we did the epitope mismatch, this is how our report now looks like. So you can put, now the physician have the 2 picture together, they can correlate the 2 items together. But actually what I found it very interesting is this part here. So because if you look at the DR beta 1, they have 1 antigen mismatch, but they have no epitope mismatch, 0. So, and if you look at the DR beta 3, it is actually the other way around. So there is no antigen mismatch because DR53 is the main antigen. But when you look at the allele level is different. So there is actually 6 epitope mismatches. And that's why I found this is very fascinating. So in the second part of the talk, I will just switch gear here. And I know that you are, all of you here are expert in immunology. And I just want to highlight something here about the indirect allo recognition, and because this is our next project. But if you don't mind, can you accept that I change the name a little bit to call it indirectly recognizable? And the idea here is, like, for example, here, if you have a donor cell, this donor cell expresses HLA. At one point, this HLA will be shed off, and then it will be taken by one of the antigen presenting, from the donor antigen presenting cells. Say, for example, this is a B cell. The B cell will recognize a specific epitope into this HLA, and then this HLA will become endocytosed into the cell. And after it becomes endocytosed, it will get fragmented into different molecules. And then at one time, this epitope will be attached to the MHC, And this is what we call HLA epitope. And the idea here is that it is a linear epitope. It could be something inside the molecule that you don't see it from the surface, but when it become degraded, now become exposed and it become expressed on the MHC class II. And when this happens, it actually activates the CD4 helper cells. And here the CD4 helper cells has two functions. Has a function, it's either to activate the T cell response through the cytotoxic cells, or it can activate the plasma cells and produce antibodies. So if we have a software that can predict what HLA is going to form this HLA epitope, then we'll be very lucky. And actually, we have a software. And now you can see that the word PIRCHE comes from predicted indirectly recognizable HLA epitope. And the idea here is the PIRCHE is different from the matchmaker, because the PIRCHE looks at the core HLA, but the matchmaker look only at the surface. And this is why this is a big difference. So based on this idea, or at least I'm just going to go through it. So actually, in order to do PIRCHE, the PIRCHE has 4 different levels. So the first thing, it subtracts the presented self-peptide, and I'm going to show you how. And then it identifies the allopeptide, and then it predicts the peptide binding affinity and the core peptide. After that, it gives a specific score. So each HLA epitope gets a score of 1. So let's see how it works here. If you have a donor HLA class 1 and a recipient HLA class 1, the first thing the PIRCHE program do, it actually extract every single possible HLA present in the donor. So here, for simplicity, I just added 8 different HLA epitopes. And then in the next stage, it start to compare the donor to the donor HLA to the recipient HLA, and then it actually groups this epitopes into self HLA epitope and allo HLA epitopes. We don't really care about self HLA epitopes because no one is going to form, because of our MHC restriction, there is no immune response going to happen against yourself. But we care more about the allo HLA epitope, which I'm going to explain it in the next slide. So now we will take the allo HLA epitope, and the idea of the PIRCHE, it can actually predict which one of these allo HLA epitope will bind specifically to the patient MHC based on MHC restriction. Like, for example, if this patient is DR 0301 and 0302, so might be this patient will have 2 of this HLA epitope will bind to the surface, and 2 of them will will not bind. And this will result in T cell mediated activation, and the patient will get a score of 2 because 2 of them out of the 4 has bind. And this is the idea of the PIRCHE, how it works. So we hypothesize that high PIRCHE score will correlate with increased likelihood T cell mediated rejection, while high epitope mismatch score will be more strongly associated with antibody mediated rejection. And the reason of this hypothesis is T cells are directly engaged in recognizable or recognizing mismatched peptides presented by the HLA molecules, while the epitope mismatch typically stimulate the humoral response leading to the donor-specific formation. So the first set of experiment that we did is just like a simple experiment to see if this idea is going to work. So we used here different locus, A, DR, and DQ, because these were the highest number of patients that developed donor-specific antibody, and we compared between the epitope mismatch between the donor-specific antibody and the no donor-specific antibody group. And you can see here that definitely if you are in the donor-specific antibody, you have higher score or higher Eplet mismatch score than the group that does not have the donor-specific antibody. And we also did the same with the PIRCHE. And actually PIRCHE completely agreed with what we have seen with the epitope mismatch. So it looks like both softwares are working together, which is not usual in science. So we took back our 616 patient cohort and we divided them into different groups. The first group is donor-specific antibody group, which was 105 patients. And then, as we know yesterday, that not every donor-specific antibody develop antibody-mediated rejection. So almost 50% of them develop AMR, and then there is 30% that develop mixed AMR and cell-mediated rejection. And then the other group was 74. These are the patients that develop just cell-mediated rejection. And then the rest of the patients did not develop rejection. And from this patient that did not develop rejection, 173 was actually proven by biopsy, because in our center, not everyone, we don't do a biopsy for everyone. We only do biopsy for patients who have increased their creatinine level by 10%. And these are the patients that we do biopsy for them, not everyone. And that's why the numbers are less than the actual patient that did not develop rejection. And now I'm not comparing donor-specific antibody, I'm comparing patients who actually develop antibody-mediated rejection. And you can see here, when we looked at the Eplet mismatch score, that patients who develop antibody-mediated rejection was significantly have higher Eplet mismatch score. And patients who did not develop, developed cell-mediated rejection, the score was not significant. But when we looked at the PIRCHE score, the picture has reversed now. So we found that the PIRCHE actually correlated more with patients that develop cell-mediated rejection, but did not correlate with patients that develop antibody-mediated rejection. And this is my final slide here, and actually this is the whole picture of why we are here. We found that regardless the type of rejection that happened, if you have a low Eplet mismatch and a low PIRCHE, you are actually doing the best, which is the blue line on the top. But if you have high epitope mismatch and high PIRCHE, you are actually doing the worst, which is the low or the black line at the bottom. And if you have a mixture of high and low, these are the group of patients in the middle that we don't know how they will work. And I think I will end my presentation with this slide, that epitope matching and PIRCHE can replace the traditional antigen mismatch for prediction of donor-specific antibody rejection and graft survival. They are very useful tool for organ allocation, as we have listened to all the nice talks yesterday, and also for post-transplant decision. And I think the key is personalized medicine, which we have listened to some of the talks from James yesterday, which is, I think this is the main idea of this epitope matching. And the last is, you can see that we started with agglutination, and now we are back to the HLA Matchmaker and PIRCHE. And at the end, I would like to thank again for listening, and thanks my team for doing all this nice work, and Genome Canada for inviting me. Thank you.
Hi, thanks really for that really interesting talk. I was wondering, can you in vitro recapitulate a good or a bad match with mixed lymphocyte reactions? Because I'm asking that question about whether you could, if you could then use an in vitro tool to model how different therapies would interface with good or bad matches.
So mixed lymphocyte, this was a technique before I was born, actually. So I don't know how it works, but we do actually flow cross match. This is the alternative for.
No, I mean, I'm not talking about using that as a diagnostic. I'm talking about, okay, if you have a patient, say, who's got a bad PIRCHE match, but you need to move forward anyway with that transplant for whatever reason, if you used a mixed lymphocyte reaction, can you read out that, not using the computer, with real biology, that you had a bad match, and then test different types of drugs that you might want to use to mitigate the T-cell mediated rejection?
I think it sounds very reasonable. It sounds like a good idea. I haven't done it, but I think it works, because if we are hypothesizing that it is the PIRCHE score is correlating with the activity of the T cells, which is true because each score actually activates a certain clone. So if you have multiple PIRCHE score, you have multiple clones of T cells are activated. So I think the idea around sounds feasible, but.
Yeah, I mean, well, fuck after, because I think
yeah, it is, yeah. Sounds like a cool idea, yeah. I don't know if anyone have done it before, but it sounds cool, yeah.
Actually, it's a great idea, Megan. We tried it about 30 years ago. And
Have the PIRCHE score 30 years.
no, but the question of the variability in response and the variability in response to medications. And there is an enormous heterogeneity between the same recipient against different donors and various recipients against the same donor. And there is an enormous variation also in the response to individual immunosuppressants. Now, whether you could characterize that sufficiently to reduce the heterogeneity, I don't know, but the principle, I think, stands, there's no question.
I have a question.
Oh, sure.
So for your reports, you actually provide the Eplet mismatch score.
Yes.
Do you provide a threshold on top of that saying high, low?
Yes.
Okay, so how does the clinical programs apply that score?
Okay, so that's a really good question. So we don't use the Eplet mismatch for allocation yet, but we use it for immunosuppressive adjustments. So if they are above the highest risk score, they got more monitoring for the trough level of tacrolimus, and also they get adjusted their tacrolimus based on I think they don't want the level to decrease by 6, which is I think it's very similar to Dr. Nicholson paper that he suggested. So our team uses it for this purpose yet, but we don't use it for allocation. Like, for example, if there are 2 patients, they might look into it, but they have not considered it yet. So the main idea is just for adjusting immunosuppressive so far.
Great, thank you.
Yeah.
Any more questions? Great. Thank you, Doctor.
Thank you.
Bye-bye.